Chapter 10, Cell Cycle and Cell Division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells, indeed of the all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to the new cell population that is formed from the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure of consisting millions of cells. 10.1 Cell cycle. Cell division is a very important process in the all living organisms. During the cell division, the cell DNA replication and the cell growth also takes place. Also, these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of the progeny cells containing intact genomes. The sequence events by which a cell duplicates its genome, synthesizes the another constituent of the cell and eventually divides into two daughter cells is termed as a cell cycle. Although cell growth means in terms of cytoplasmic increase is a continuous process. DNA synthesis occurs only during the one specific stage in a cell cycle. The replicated chromosomes, that is DNA, are then distributed to the daughter nuclei by complex series of events during the cell division. These events are themselves under genetic control. 10.1.1 Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by the human cells in a culture. These cells divide once in approximately every 24 hours. Figure 10.1. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases, interphase and M phase, means mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is the significant to note that in the 24-hour of average duration of a cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, that is karyokinesis, and usually ends with the division of the cytoplasm, that is cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for the division by undergoing both the cell growth and the DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase, that is gap 1, S phase, that is synthesis, and G2 phase, that is gap 2. G1 phase corresponds to the interval between the mitosis and the initiation of the DNA replication. During the G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S, or a synthesis phase, marks the period during which the DNA synthesis or a replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as a 2C, then it increases to the 4C. However, there is no increase in the chromosome number. If the cell had diploid or a 2n number of chromosome at the G1, even after the S phase, the number of chromosomes remains the same, that is 2n. In animal cell, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for a mitosis while the cell growth continues. Some cells in adult animals do not appear to exhibit division, that is hard cells, and many other cells divide only occasionally as needed to replace the cells that have been lost because of the injury or a cell death. These cells that do not divide further exit the G1 phase to enter the inactive stage called the quiescent stage, G0, of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where the haploid cells divide by mitosis. For example, male honeybees. Against this, the plant cells show the mitotic division in both haploid and the diploid cells. From your recollection of examples of alteration of generations in plants, that is chapter 3, identify the plant species and the stages at which the mitosis is seen in haploid cells. 10.2 MPhase. This is the most dramatic period of the cell cycle involving a major recognition of the virtually all components of the cell. Since the number of chromosomes in the parent and the progeny cells is the same, it is also called as a equational division. Though for the convenience, mitosis has been divided into 4 stages of nuclear division, that is karyokinesis, it is very essential to understand that the cell division is a progressive process and very clear-cut lines cannot be drawn between the various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase and telophase. 10.2.1 Prophase. Prophase, which is the first stage of the karyokinesis of mitosis, follow the S and G2 phases of the interphase. In the S and the G2 phases, the new DNA molecules formed are not distinct but intervene. Prophase is marked by the initiation of the condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. Figure 10.2a. The centrosome, which had undergone duplication during S phase of the interphase, now begins to move towards the opposite pole of the cell. The completion of the prophase can thus be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosome. Chromosomes are seen to be composed of the 2 chromatids attached together at the centromere. Centrosome, which had undergone duplication during the interphase, begins to move towards the opposite pole of the cell. Each centrosome radiates but microtubules called asters. The 2 asters together with the spindle fibers forms the mitotic apparatus. Cells at the end of the prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, nucleolus and the nuclear envelope. 10.2.2 Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of the mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of the chromosome is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of the chromosome is most easily studied. At this stage, metaphase chromosome is made up of 2 sister chromatids which are held together by the centromere. Small disc-shaped structures are at the surface of centromeres are called kinetochores. These structures serve as the sites of attachment of the spindle fiber to chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by the chromosomes coming to lie at the equator with the 1 chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fibers from the opposite pole. Figure 10.2b. The plane of alignment of the chromosomes at the metaphase is referred to as the metaphase plate. The key features of metaphase are spindle fibers attached to kinetochores of the chromosomes, and chromosomes are moved to spindle equator and gets aligned along the metaphase plate through the spindle fibers. 10.2.3 Anaphase. At the onset of the anaphase, each chromosome arranged at the metaphase plate is split simultaneously and the 2 daughter chromatids, now referred to as the daughter chromosomes of the future daughter nuclei, begin their migration towards the 2 opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence leading the edge with the arms of the chromosome trailing behind. Figure 10.2c. Thus, the anaphase stage is characterized by the following key events. Centromeres split the chromatids separate. Chromatids move to the opposite poles. 10.2.4 Telophase. At the beginning of the final stage of the karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each set of the chromatin materials tend to collect at each of the two poles. Figure 10.2d. This is the stage which shows the following key events. Chromosomes cluster at the opposite spindle poles, and their identity is lost as a discrete elements. Nuclear envelope develops around the chromosome cluster at each pole, forming the two daughter nuclei, nucleolus, Golgi complex, and ER reform. 10.2.5 Cytokinesis. Mitosis accomplishes not only the segregation of the duplicated chromosomes into the daughter nuclei, that is karyokinesis, but the cell itself is divided into two daughter cells by separation of the cytoplasm, called the cytokinesis, at the end of the which cell division gets completed. Figure 10.2e. In an animal cell, this is achieved by the appearance of the furrow in the plasma membrane. The furrow gradually deepens and ultimately joins the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by the relatively inextensible cell wall. Therefore, they undergo the cytokinesis by a different mechanism. In plant cell, wall formation starts in the center of the cell and grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of the simple precursor called the cell plate that represents the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by the cytokinesis, as a result of which the multinucleate condition arises, leading to the formation of the syncytium. Example, liquid endosperm in the coconut. 10.3 Significance of the mitosis. Mitosis or equational division is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, haploid cells also divide by the mitosis. It is very essential to understand the significance of the division in the life of an organism. Are you aware of some examples where you have studied about the haploid and diploid insects? Mitosis usually results in the production of the diploid daughter cells with the identical genetic complement. The growth of a multicellular organism is due to mitosis. Cell growth results in the disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of the mitosis is a cell repair. The cells of the upper layer of the epidermis, cells of lining of the gut, and the blood cells are being constantly replaced. Mitotic divisions in a meristematic tissues and the apical and the lateral cambium result into the continuous growth of the plants throughout their life. 10.4 Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with the complete haploid set of the chromosomes. Gametes are formed from the specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half result in the production of the haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of a sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during the gametogenesis in the plants and animals. This leads to formation of the haploid gametes. The key features of the meiosis are as follows. Meiosis involves the two sequential cycles of the nuclear and the cell division called the meiosis 1 and meiosis 2, but only the single cycle of a DNA replication. Meiosis 1 is initiated after the parental chromosomes have replicated to produce the identical sister chromatids at the S phase. Meiosis involves the pairing of homologous chromosomes and the recombination between the non-sister chromatids of the homologous chromosomes. 4 haploid cells are formed at the end of the meiosis 2. Meiotic events can be grouped under the following phases. 10.4.1 Meiosis 1. Prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to the prophase of the mitosis. It has been further subdivided into following 5 phases based on the chromosomal behavior, that is liptotin, zygotin, pectin, diplotin and diakinesis. During the liptotin stage, the chromosomes become gradually visible under the light microscope. The compaction of the chromosome continues throughout the liptotin. This is followed by the second stage of the prophase called zygotin. During this stage, chromosomes start pairing together and this process of association is called the synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that the chromosome synapsis is accompanied by the formation of complex structures called the synaptonemal complex. The complex formed by the pair of synapsed homologous chromosomes is called the bivalent or a tetrad. However, these are more clearly visible at the next stage. The first two stages of the prophase 1 are relatively short-lived compared to the next stage, that is pectin. During this stage, the 4 chromatids of the each bivalent chromosomes become distinct and clearly appear as a tetrads. This stage is characterized by the appearance of the recombination nodules, the site at which the crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of the genetic material between two homologous chromosomes. Crossing over is also an enzyme mediated process and the enzyme involved is called the recombinase. Crossing over leads to the recombination of a genetic material on the two chromosomes. Recombination between the homologous chromosomes is completed by the end of the pectin, leaving the chromosomes linked at the site of the crossing over. The beginning of the diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosome of the bivalents to separate from each other except at the site of the crossovers. These X-shaped structures are called the chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of the meiotic prophase 1 is a diakinesis. This is marked by the terminalization of the chiasmata. During this phase of the chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosomes for a separation. By the end of the diakinesis, the nucleolus disappears and nuclear envelope also breaks down. Diakinesis represents the transition to the metaphase. Metaphase 1, the bivalent chromosome align on the equatorial plate, figure 10.3. The microtubules form the opposite poles of the spindle attached to the kinetochore of the homologous chromosomes. You can see the stages of meiosis 1. Anaphase 1, the homologous chromosomes separate while the sister chromatids remain associated at their centromeres, figure 10.3. Telophase 1, the nuclear membrane and the nucleolus disappear. Synatokinesis follows. This is called as a dyad of the cells, figure 10.3. Although in many cases the chromosomes do undergo the some dispersion, they do not reach the extremely extended state of the interphase of the nucleus. The stage between the two meiotic division is called the interkinesis and is generally short-lived. There is no replication of DNA during the interkinesis. Interkinesis is followed by the prophase 2, a much simpler prophase than the prophase 1. Meiosis 2, prophase 2. Meiosis 2 is initiated immediately after the cytokinesis, usually before the chromosomes have fully elongated. In contrast to the meiosis 1, meiosis 2 resembles to the normal mitosis. The nuclear membrane disappears by the end of the prophase 2, figure 10.4. The chromosomes again become compact. Metaphase 2. At this stage, the chromosome align at the equator and the microtubules from the opposite poles of the spindle get attached to the kinetochores, figure 10.4 of the sister chromatids. Anaphase 2, it begins with the simultaneous splitting of the centromere of the each chromosome which was holding the sister chromatids together, allowing them to move towards the opposite pole of the cell, figure 10.4, by shortening of the microtubules attached to the kinetochores. You can see the stages of the meiosis 2. Telophase 1, telophase 2. Meiosis ends with the telophase 2, in which the two groups of the chromosomes once again get enclosed by the nuclear envelope. Cytokinesis follows, the resulting in the formation of the tetrad of the cells, that is 4 haploid daughter cells, figure 10.4. 10.5 Significance of the meiosis. Meiosis is the mechanism by which the conversion of the specific chromosome number of each species is achieved across the generation in sexually reproducing organisms. Even though the process per se paradoxically results in the reduction of the chromosomes number by half, it also increases the genetic variability in the population of organisms from one generation to the next. Variations are very important for the process of evolution. Summary. According to the cell theory, cells arise from the pre-existing cells. The process by which this occurs is called the cell division. Any sexually reproducing organism starts its life cycle from a single celled zygote. Single cell division does not stop with the formation of the mature organism but continues throughout its life cycle. The stages through which the cell passes from the one division to the next is called the cell cycle. Cell cycle is divided into two phases called the interphase, a period for preparation of cell division, and two, mitosis, means M phase, the actual period of the cell division. Interphase is further divided into G1, S and G2. G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occurs during this phase. S phase marks the phase of DNA replication and chromosome duplication. G2 phase is the period of the cytoplasmic growth. Mitosis is also divided into four stages, namely prophase, metaphase, anaphase, telophase. Chromosome condensation occurs during the prophase. Simultaneously, the centrioles move to the opposite poles and the nuclear envelope and the nucleolus disappears and the spindle fiber starts appearing. Metaphase is marked by the alignment of the chromosome at the equatorial plate. During the anaphase, the centromeres divide and the chromatid starts moving towards the two opposite poles. Once the chromatid reach to the two poles, the chromosomal elongation starts. Nucleolus and the nuclear membrane reappear. This stage is called telophase. Nuclear division is then followed by the cytoplasmic division and is called the cytokinesis. Mitosis thus is the equational division in which the chromosome number of the parent is conserved in the daughter cell. In contrast to the mitosis, meiosis occur in the diploid cells, which are destined to form the gametes. It is called the reduction division since it reduces the chromosome number by half while making the gametes. In the sexual reproduction, when the two gametes fuse, the chromosome number is restored to the value of the parent. Meiosis is divided into two phases, meiosis 1 and meiosis 2. In the first meiotic division, the homologous chromosomes pair to form the bivalents and undergo crossing over. Meiosis 1 has a long prophase which is divided further into 5 phases. These are liptotin, zygotin, pectin, diplotin and the dikinesis. During the metaphase 1, the bivalents arrange on the equatorial plate. This is followed by the anaphase 1 in which the homologous chromosome move to the opposite poles with the both of their chromatids. Each pole receives half the chromosome number of the parent cell. In the telophase 1, the nuclear membrane and the nucleolus reappear. Meiosis 2 is similar to the mitosis. During the anaphase 2, the sister chromatids separate. Thus, at the end of the meiosis 4 haploid cells are formed. Thank you.